So, at this point, we refer to the RNA sequence as quote-unquote pre-mRNA, but once it's ready to leave the nucleus and be translated, we will refer to it as messenger RNA, or just normal mRNA. The first modification that takes place is a 5' cap is added to the 5' end of the pre-mRNA strand. While DNA is being transcribed into RNA, a 7-methylguanosine cap is added to the 5' end of the growing RNA transcript by a phosphate linkage. This addition of this phosphate group is important because it protects the newly forming RNA from degradation. It also helps with protein synthesis, or more specifically, translation, because factors that are involved in translation recognize this cap and help initiate translation by the ribosome. The next modification that must be made is the addition of a poly-A tail to the 3' end of the strand. After the elongation step of transcription, an enzyme called poly-A polymerase adds a string of approximately 200 adenines to the 3' end of the RNA transcript. We call this series of 200 adenines the poly-A tail. And the poly-A tail further protects the pre-mRNA from degradation, and it is also the binding site for a protein necessary for exporting the processed mRNA out of the nucleus and into the cytoplasm. Now, something wonky about eukaryotes is that their genetic code actually contains a lot of quote-unquote extra information. So to make sure only the important bits leave the nucleus and are translated into proteins, something called RNA splicing must also occur before the mRNA strand can leave the nucleus. Eukaryotic genes are made up of exons, which correspond to protein coding sequences, and intervening sequences called introns. The X in the word exon signifies that they are the part of the gene that gets expressed, while the int in introns denotes their intervening role in the sequence. Intron sequences in mRNA do not encode functional proteins. Introns must be removed to make a translatable mRNA. The process of removing introns and then reconnecting the exons is called splicing. The splicing process is catalyzed by protein complexes shown on the screen in purple called spliceosomes. Spliceosomes are composed of proteins, which are easily seen on the screen here, and RNA molecules that are too small to be seen here, but are associated with these proteins, small nuclear RNAs, or snRNAs, because they are small bits of RNA that reside in the nucleus, so small nuclear RNA. The beginning and end of each intron is marked with specific nucleotides. GU at the 5' end and AG at the 3' end of the intron. Spliceosomes recognize these sequences at the 5' and 3' ends of the intron, so they know where to make cuts to remove the intron from the sequence. So, to reiterate, transcription occurs within the nucleus. After DNA is transcribed into RNA, before the RNA can leave the nucleus, the RNA must first, 1. Have a 5' cap added to the 5' end, 2. Have a poly-A tail added to the 3' end, and 3. All of the introns must be removed from the RNA sequence through a process called splicing. After all of this is complete, we now have an mRNA strand that is ready to leave the nucleus and be translated into a protein.